All right, so this is the final section of notes on 11.2, movement. Today we're going to be looking at the mechanism of the actual contraction and relaxation within the muscle. To understand this, you might want to take a moment, go back and review all of your structures, vocabulary, regain perspective, looking at where all of the stuff is going to be taking place. All right, so let's talk about the mechanism of muscular contraction. First of all, this is going to take place kind of on two levels. We're going to start off by looking at the myofiber or the muscle cell, and then kind of take it down to the sarcomere level within the myofibrils. Okay, so the first thing that's going to happen is a neurotransmitter is going to be received at the myofiber level. This is going to cause depolarization or an action potential of the myofiber membrane. Now that membrane is called the sarcolemma, and again, they do not have those finger-like projections like dendrites and neuron cells typically have. So we call this the neuromuscular junction. So it's where the message from the neurons is going to be transmitted over to the muscle fiber. The process from the neuron end is identical. You have the neurotransmitters, moving across that synaptic cleft, and then it's going to be received by receptors, neurotransmitter receptors in the sarcolemma or the plasma membrane of the muscle cell. Okay, so you may recall that calcium ions were really important in neuron transmission. So as soon as this action potential is reached or received by the muscle cell, it's going to release calcium ions. Calcium ions are super important. They play a number of different roles, and so we want to look at these in more detail. All right, so the first thing that's going to happen is once those calcium ions are received, they are triggered by the reception of the neurotransmitter, these calcium ions are going to flood down into the myofibrils through a little structure called the T-tubule. You don't actually have to know that. That is going to release trigger the release of calcium ions from the sarcoplasmic reticulum. So the sarcoplasmic reticulum is inside the myofiber or muscle cells, and it's surrounding each one of those little myofibrils. So envision sarcomeres bundled together inside the myofibril, and then the sarcoplasmic reticulum around that, all within the muscle cell. Okay, so this sarcoplasmic reticulum is highly specialized. It has tons of calcium, and its main job is to release and then reabsorb calcium ions. All right, so now on the myofibril level, the calcium ions are going to bond to that troponin. Now remember, the troponin is on the actin fibers along with the tropomyosin. So now we're down to the sarcomere level with the actin and myosin combination there. So once the calcium ions bond to the troponin, they're going to cause a shape change in the troponin. When that happens, the troponin pulls on the tropomyosin and exposes those myosin receptors, or the little myosin head connectors, so to speak, on the actin. All right, so this kind of shows you a quick diagram. We've looked at this before, so you see the axonic bulb up above, and so what I want to point out here, I'm not going to spend a lot of time talking about it, but again, this right here is going to be the sarcolemma, and that is simply the plasma membrane of the muscle cell. So the myofibrils are inside here. So here's your neuromuscular junction or complex. So again, this is kind of showing what would be a dendrite on another neuron. So you can see we have the acetylcholine receptor sites. Those are usually on dendrites on neurons, but with the muscle cells, they're just little, embedded proteins. in that sarcolemma or plasma membrane of the muscle cell. All right, so now what's going to happen is those on the actin fibers, the places where the myosin can actually, bond onto the actin fibers, so the myosin binding sites, they are now exposed because we moved that tropomyosin out of the way. So now the little myosin heads can bond to the myosin binding sites on the actin. This forms what we call a cross bridge. If you go back and look at your assessment statements, there's one point where it says you need to show the myosin heads attached to the myosin binding sites or receptors, and that is the cross bridge component. So we have the myosin down here, and then of course here is the actin. Okay, so again, calcium ions were triggered by the reception of the acetylcholine. They flood down into the muscle cell. Each myofibril is covered with sarcoplasmic reticulum. It's going to be triggered to release even more calcium ions. The calcium ions are going to come and bond to the troponin. The troponin will pull on the tropomyosin. That exposes the myosin binding sites on the actin. Now the myosin heads are going to pop up and form this cross bridge. We'll get into that in more detail in just a second. All right, so here's basically how it happens. There is an ATP molecule attached to the myosin head. It is going to be hydrolyzed to break down into ADP, the lower energy form, the phosphate that was broken off, and the energy. So remember, there's energy contained in the covalent bond between the second and third phosphates in the ATP molecule. So when we hydrolyze it and we break that bond, we release that energy. Law of conservation of energy, energy went into hooking that third phosphate on, and it was stored in the bond. When we break it off, we release that energy back out. Okay, so that energy is actually stored in the myosin head, so it's now energized. The ADP and the phosphate that was broken off, they're now separated. They remain attached at this point to the myosin head. So as I said, the energy that was released from the covalent bond between the second and third phosphate is now going to be stored in the myosin head. And it is actually used to set or cock the myosin head. So it kind of sets it, and it's cocked at this funny angle, so it's just ready to pop up and attach to the actin at the myosin binding sites. All right, so now the myosin heads are going to bind to the receptors of the myosin binding sites on the actin. Again, this is going to form the cross bridge. So this can't happen without the hydrolysis of ATP and the energy that is produced when ATP is broken down. Now we have the power stroke, and we'll watch some animations of all this in just a second. The power stroke is where the myosin head is going to pop up or extend. It kind of pivots, almost like if you hold your elbow in one position and then hold your forearm up and you swing your arm back and forth. So that's kind of what the myosin heads do. Remember, at this point, they are attached to the actin filaments by the myosin binding sites on the actin. They are going to slide the actin fiber toward the M line, or the center of the sarcomere. So this is going to shorten the sarcomere. When you have this happening simultaneously all along multiple sarcomeres, that's going to shorten the muscle. Okay, so now ADP and the phosphate are going to be released. Okay, so when ATP comes in and reattaches to the myosin head, that is going to detach it And so this is when we get the cell starting to relax. So if you think about what's happening here, ATP is going to be used to actively transport the calcium ions back into the sarcoplasmic reticulum, back on the actin fiber. So once the sarcoplasmic reticulum has reuptaked, not very good grammar there, has taken the calcium ions back in, think about what's going to happen there. The troponin is going to relax. That's going to put the tropomyosin back into its original position. That's going to cover the myosin binding sites or myosin receptors. And so at that point, the muscle cell is relaxed. The myosin is detached from the actin. It can't ratchet it along. So finally, the ATP is going to hydrolyze. This is the beginning of the next cycle, or a continuation of a previous contraction if the cell does not actually relax in between. The ATP is going to hydrolyze. It's on the myosin head. It's broken down into ADP and phosphate, and the energy that was released stored in the myosin head. That recocks it. Now the whole cycle is ready to continue. All right, go ahead and stop here, and then we will watch the animations together in class if you're present. If this is makeup because you were absent, strongly recommend that you stop here and go ahead and watch these.